right, so we'll talk a little bit about scientific inquiry today and how scientists learn things. And a lot will be reviewed from 7th grade and 8th grade probably, but we're going to get into some more detail later on in this section that we didn't really get into, and talk a little bit more about the nuances of the scientific method and some things related to that. Anyone know who that is? Yeah, Jane Goodall. Well, you don't have him, right? I don't remember what this picture's from. I think it's Rosalind Franklin. Discover structured DNA. All right, so what is science? Well, you could sort of look at science as kind of, you can sort of look at science as a couple things. Could view science as a body of knowledge. You know, this book, this textbook of biology, is a bunch of information scientists have learned over the last couple hundred years about living things. So science is a bunch of concepts that scientists have learned. But science is also a way of learning. So when we talk about scientific inquiry, what we're talking is how do scientists learn about the world? Okay? Science is a philosophy about how you can gather information. A process that helps to answer questions about the natural world. So that's kind of what we're talking about here. So how do you do that in science? What do scientists do to learn about the world? Well, there's a couple different ways that scientists do this. There's many ways, I should say, not just a couple. One way that scientists learn about the world is through something called discovery science. So it's a little different than probably what you think of in terms of experimentation. In discovery science, basically scientists look at data and try to find patterns within that data. So especially important nowadays where we have huge data sets available that can be analyzed using machine learning, using artificial intelligence, using data analytics, where we take huge amounts of data, sort of look at it, try to find patterns using mathematics that can help us understand the natural world. That's opposed to science in which we're hypothesis testing, which before we have data, we're thinking about the problem, formulating a hypothesis, and then collecting data to test it. Okay? But they're both different types of, different ways of approaching science. So in hypothesis testing, this is the one we're probably familiar with that we've talked about lots of times. One way to go about doing this is to start by asking a question. That's what we were doing in our slug lab. We're following this process. You ask the question, what kind of foods do slugs prefer? And that's a question we could try to answer using an experiment. That's what we did. And so after you have a question you're asking, we start with a hypothesis, a prediction. Before you do your experiment, come up with your hypothesis, what you think, based on what you know about the organisms, based on what you know about the topic, you make a prediction. Test that prediction using an experiment. You set up a way to test and see is your prediction correct or not. That's an experiment. And when you gather the results of your experiment, when you're collecting data, making observations, what you're doing is you're comparing what you found in the experiment to what you thought beforehand in your hypothesis. And you see, does the data I collected agree with my original hypothesis? That's your conclusion. Sometimes it does. Sometimes your hypothesis is confirmed. The data supports what you said beforehand. Other times, the data might not support what you thought. Okay, your hypothesis that you came up with might be rejected. Just out of curiosity, for the slugs, how many people that hypothesis was supported by the data? And rejected? About half and half. Now, If your hypothesis is incorrect, if it was rejected by your data, it doesn't mean you did something wrong. It doesn't mean you should change it. You still have learned something valuable from it. And then this process sort of repeats. Your hypothesis was confirmed, you might ask another question about the topic. Try to learn some more. Your hypothesis was rejected, you might come up with an alternative hypothesis to test and repeat the process again. Now this is what we might call the scientific method, but really there isn't a single scientific method. A single set of steps that a scientist goes through and follows. Often we talk about it that way, especially at the younger age levels, like when you guys are in 7 years old. Here's the scientific method. You start with a problem and research. Well, it doesn't really work that way. Some scientists might follow that set of steps, others might not. You do things in a variety of different ways. But there are some common threads in how scientists approach a problem. And that's what we're talking about here. Scientists ask questions, lots of questions. And the questions that scientists ask are questions that can be answered in experiments. So some examples, and we're going to use these examples as we go through all these different parts. How does the amount of nitrogen fertilizer affect the height of pea grains? That's a good scientific question. We could answer that using a pretty simple experiment. Does increasing the temperature of an enzyme increase its rate of reaction? Do high doses of vitamin C prevent people from getting the common cold? These are all questions that we could ask, and we could find an answer using scientific inquiry. Once you have a question, then you generate a hypothesis, a prediction about what's going to happen, about the outcome of the experiment. So there's some important rules to a hypothesis. It's a statement. It's a prediction. It can never be a question. It has to be testable. It has to be falsifiable. It means that you could conceivably disprove this hypothesis using an experiment. It's not a rule, but often we will write our hypothesis as in the form of an if-then statement. If the independent variable changes in some way, then the dependent variable will change in this way. It's a good way to structure a hypothesis because it makes sure that you have both parts. So here are some examples. If pea plants are given additional nitrogen, then they will grow taller. That has the independent variable, the amount of nitrogen. It has the dependent variable, the height of the plant. If the temperature of salivary amylase is increased to 40 degrees Celsius, then the enzyme activity will decrease. Again, it has both parts. The independent variable is the temperature, the dependent variable is the enzyme activity. Here's one that's not phrased as an if-then, but it's still a correct hypothesis. Individuals that are given 200% of the RDA of vitamin C would be less likely to contract the cold. I can test that, I can set up an experiment. Has both parts, it just isn't phrased as an if-then. So we ask a question, we create a hypothesis, and then we have to test it. We test it using an experiment. In science, we use controlled experiments, where we're sort of very carefully trying to control as many factors as possible. In order to test our hypothesis. So in a controlled experiment, we have at least two groups, often more. We need a control group, and then we need experimental groups. The control group is kept under what we might say are normal or typical conditions. We're not altering anything in the control group. Kind of just let it be the way it would normally be. think about the pea plant fertilizer experiment, pea plants that are given just water, no additional fertilizer. They would serve as a control group. And then the experimental group, we treat them in some way. We're changing the independent variable. So the pea plants in which we're giving additional nitrogen, they are the experimental groups. You might have several experimental groups. We need that control group as a comparison. In order to learn in our experiment, we have to be able to compare the experimental group to something else, to normal conditions. That's why we have that control. All right, we have variables in our experiments. We have independent variable, we have dependent variables, we have variables we hold constant. Who can tell me what would be a definition of independent? Thing you're altering, the thing you're changing, the thing you're testing. Factor that you're manipulating in order to see how it affects the sample. A good experiment has just a single independent variable. There's just one thing different between your groups. So the examples from our experiments we were just talking about, the amount of nitrogen we give the pea plants would be the independent variable. The temperature that we're keeping the enzyme at, that's the independent variable. The amount of vitamin C we give people, that's the independent variable. We're altering each of those things. And we're seeing how they affect what? How the independent variable affects, Grace? The dependent variable. The dependent variable, we think, depends upon the independent variable. That's why it's called that. It's the data we're collecting, the results of our experiment. For example, the height of the pea plants, the amount of enzyme activity, the number of people that get the cold. Those results are our dependent variable. And then we have the variables being held constant. We only want one thing different between our control and experimental groups, where there must be a whole bunch of things that are kept the same. Sometimes these are called controlled variables. The amount of soil that we are growing the pea plants in, the amount of water we are giving, the variety of pea plant. Those are all things I would want to keep the same in that experiment. With the enzyme, I would want to keep the pH of the solution the same. The vitamin C, I would want the age of the people in the experiment to be the same, the diet the same, maybe the same test subjects, the health of the test subjects. Those are all variables I would try to keep constant. Once we have our experiment set up, then we will be making observations about it, collecting data, analyze that. Sometimes we might do it just once, like we might just measure the pea plants at the end of the experiment. Other times you might measure several times throughout the experiment. Maybe you measure their height every day for 2 weeks. Sometimes we have quantitative observations, which are numerical, they're measured, they have a number. Other times we may have qualitative measurements that don't have a number, they're descriptive. We usually collect our data in a data table, an organized way of keeping our data clear. But a large data table is often not easy to sort of find trends and so forth. So we might graph our data to see if any trends emerge that we can see. We also often will have to, some examples. We measure the pea plant's height every 3 days. That would be observations we're taking. We measure how much gas the enzyme is producing. We determine how many people get a cold during our experiment. Those are examples of observations we would be taking. And then eventually, once we have collected our data, we have to make a conclusion. We have to analyze our data and determine, does this data support our hypothesis, or should our hypothesis be rejected based on our data? We do this by comparing the control group and the experimental groups. Now there's a question, because we might do an experiment. Let's say that I repeated the slug experiment and made it better. And I have 50 slugs in separate containers. Each one got watermelon and cucumber. When I collected data from all 50 of those slugs, the average slug ate 48% of the watermelon. The average slug ate 50% of the cucumber. Would that be a good conclusion then, that slugs prefer cucumbers? What do you think? Is there a difference in our observations? Yeah, 50% of the cucumber was eaten on average and 48% of the watermelon. Does that mean slugs prefer cucumbers? Is that enough of a difference? Could be. Or not. It could just be due to random chance. They happen to randomly eat 2% more cucumber than watermelon. So how do we know if it's enough of a difference? What if they ate 10% watermelon and 50% cucumber? You could be more confident in that case. But we actually have some mathematics we use, some statistics that allow us to say, I'm 95% sure that slugs prefer cucumber versus watermelon based on the results of this experiment. And this week we're going to be talking about one of those statistical tests that helps us determine, is this enough of a difference for me to make that conclusion with 95% confidence? So we have to analyze our data. We have to do some mathematics to understand those probabilities. So some examples of conclusions we might make: if the pea plants given fertilizer grew an average of 8.3 centimeters taller than the control plants, my hypothesis would have been supported. If the salivary amylase was 40 degrees Celsius, had 23% lower enzyme activity, hypothesis supported from that data. Or I might find there's no significant difference in the number of people contracting a cold that were given more vitamin C. Hypothesis rejected. So that word significant becomes important. You're almost always going to find a difference in the number between two groups. But is it a significant difference? That's where statistics are. If our hypothesis is supported, we might repeat the experiment to confirm. If you repeat the experiment and you get a different result, well then we need to look at that experiment. We made sure our experiment's valid, make sure we controlled for all the variables. In science, when scientists learn something new from an experiment, from some research, they share that with other scientists. And they do that through the process that's called peer review. In peer review, basically, scientists will write up their experiment that they conducted, very clearly explain all the procedures, show a summary of their data, talk about their conclusions, and then they will submit it to a journal, they're called. And that journal will send it around to a few other scientists. They'll read it over. They might edit, they might ask questions about it. If they agree that it was a good study, they will then publish it, basically in a magazine. And people subscribe to journals that are appropriate to their field. If you were a researcher studying, corn viruses, you might subscribe to a journal of people researching corn plants. Every month you get in the mail a magazine about this thick that has 7 or 8 different research studies in it. You might read about them. You would learn what other scientists in your field are doing. If they found some new information, you would then be able to understand that. You might get ideas from that research. You might repeat their experiments and confirm it. That's what peer review means, sharing and publishing the results. Tomorrow you're going to be reading a scientific article, an actual scientific paper published in the journal Science, which is probably one of the most well-known scientific journals. It covers all different areas. It's not just specific to one content area. And you'll see what a peer-reviewed scientific paper looks like, and start to look at that. When scientists read about this research, like I said, they might ask new questions, they might repeat the process again. Other times a hypothesis that was arrived at, or that was formulated, might be rejected. The data might not agree with it. Okay, that's okay. It's still valuable. Might lead to other ideas, other hypotheses. Might repeat the experiment with a different idea. So whether the hypothesis is supported or rejected, it's still of value. So what do we need for a valid experiment? When we say valid in an experiment, we can trust the results. We can be confident in what this experiment has told us. Well, we need as many samples as possible. The more samples we have in our experiment, the more confident we can be. Our slug experiment is not a great one in terms of sample size. If you have a small sample, if you did an experiment saying, okay, does vitamin C help people not get a cold? And you only have 5 people in your group, your research group, that's not very many. And due to random chance, 3 of them may have gotten a cold because of who they were hanging around with. The more samples you have, you can sort of ensure that results are less likely due to chance. Okay? There could be outliers in your experiment, data points that are sort of much different from others just due to random chance. The more samples you have, the less effect an outlier is going to have on the data. A valid experiment has one single independent variable as much as possible. We try to control all the other variables if we can. It's almost impossible to really control everything. We do the best we can to control those other variables. We repeat an experiment to confirm the results, to see if what we found the first time can be found again in a second. All right, so let's think about this and just try to identify some of the parts of this experiment. So if we conduct an experiment with 30 pea plants, 10 are given no fertilizer, 10 are given 5 grams of fertilizer, 10 are given 15 grams of fertilizer. We grow them for 14 days and then measure the height of the plant. In that experiment, what would be a possible hypothesis that we're testing if we were to set that up? Priscilla?
If you give more fertilizer, the plants will grow.
Okay, so yeah. If plants are given extra fertilizer, then they will grow taller. It has both parts of my hypothesis. Has the independent variable and the dependent variable. It's testable, it's clear. So what is the independent variable? If that's the experiment we're doing, that's our hypothesis. What would our independent variable be in this experiment? Rita? The amount of fertilizer. That's what's different between my two groups. They would be giving different amounts of fertilizer. What would my dependent variable be in this experiment? Emma? The height of the plants. That's what I'm measuring. That's the data I'm collecting. That's the results. Those are my observations. What would be some control variables? Some variables I would want to hold constant, right? Yeah, the amount of water they're getting, the amount of sunlight. Do you have a different one, Carter? The environment they're in. Yeah, there could be lots and lots of things you might think about. Dustin. What would the control group be in this experiment? I'd love to hear from some new participants. Zach?
No fertilizer plants.
Yeah, the plants that were grown without fertilizer. What about the experimental groups? Grady?
Plants with 15 grams or less.
Yep. And the ones with 5 grams as well. Both of those groups are experimental. You often will have several experimental groups getting different levels of treatment in an experiment. How can we improve this experiment? This is an easy answer to this question. This is a common question, like on the Regents exam. You can almost always answer it the same way. How could this experiment be improved? Grace? Do it several times, or come on, larger sample size. We'll hold off on talking about graphing until later in the week when we address that. You guys seen Monty Python and the Holy Grail movie? Yes. Are you fans? Let's watch this thing. We only got a few minutes.